Hello, welcome. Top of the Talktoptera to you, Tucker and Buck. It's May 19th, 2025, and we're in the first Talktoptera with an old entomological colleague and friend, Buck. So, Tucker, first, if you wanted to say hello, please, and then we will pass to Buck to get things started.
Hi, I'm Tucker, longtime insect and ant enthusiast, occasional research participant, and here to help push the conversation on ants and insects further and further afield. So, looking forward to hearing from Mr. Trible about his research.
Cool.
Okay.
So, Buck, what's your background? How did you get started, and what's your research focus, and how's it developed over the last couple of years?
So I'm kind of a ant genetics lifer. I've been studying sort of the genetics and evolution of ants since maybe 2010 when I was an undergraduate in college. Um, and yeah, so I've had a somewhat traditional academic career, went from my undergrad to a PhD. Now I'm running a small lab at Harvard as like an independent research fellow. Um, and I'm just about to wrap up my time at Harvard and move to a faculty position at the University of Georgia.
Georgia, America.
Yeah.
Okay, that's exciting. What got you into ants in your undergrad career? Was it just because there was an interesting opportunity, you liked the specific mechanism, or was it the ants that made you forge that direction?
Um, so I think that my early career was I was always very much interested in nature, being outside. I grew up in a more rural area in Virginia, and my family did a lot of hunting and spending time outdoors, and so there was a lot of just emphasis on the natural world my whole childhood. And um, at a certain point I got really interested in evolution, um, classic route to that, reading things like Neil Shubin or Richard Dawkins, kind of popular books about evolution that I read maybe in high school. um, and I had this opportune thing where I was a counselor at a summer camp right before I went to college, and I was teaching a course on entomology. So it was like a sort of like a science camp for kids, and you're teaching short sessions. And I was just given course materials to teach, but the topic I was sort of randomly given to teach was entomology, and I just totally fell in love with the
Yeah,
topic.
what a gift.
And particularly from the perspective of evolutionary biology, the thing about entomology and insects is that it feels like you can really see evolution happening. Like you see all of these different forms, you see these kind of gradations and different evolutionary strategies, and it all kind of really clicks in a way that is more abstract when you're studying other types of organisms.
Yeah, totally. And I mean, Dan and I talk about this often, but insects and ants really offer this glimpse into like a micro scale because they're quite small and they occupy very small niches, and they bump up against the macro scale. And that's coupled with the fact that with social organisms like ants and termites and bees, you have this interesting relationship between the individual, we'll call it like a cellular structure, and then an organism, a larger structure. You
Sure.
can actually see it with your eyes. You can go with a microscope and look at cells and tissues, but ants and insects give you like a palpable view of evolution and with their
Totally.
speed of development. And this actually touches on a lot of your research that you've done with single-step, large phenotypic evolution that you've seen in ants via genetic manipulation and CRISPR, which I'm sure we'll get into. Dan, do you have any questions as we move along into maybe your master's research and then your PhD research and then ultimately how you run your lab?
Great. No, we will definitely get to the mute ants and all that happened since then. Yeah. Continue however you want, Buck.
Well, just to say that, yeah, of course, yeah, like my joke used to be that like if you're interested in evolution, uh, you should study insects. I mean, like evolution genetics, of course, being deeply intertwined topics, right? And the basic understanding of genetics that we have, like the earliest history of it, started with things like bacteria or phages, but then it quickly jumped to fruit flies, and fruit flies really were, and in some ways remain, the workhorse of our understanding of how genetics works. I mean, that's where chromosomes were discovered and all kinds of things, right? And so my joke used to always be that like if you're interested in evolution, you should study insects, and if you're interested in insects, you should study ants because they're the best ones.
Yeah.
And of course, there's lots of incredible insects, but um, it's sort of like a um, yeah, it's almost like a conversion experience or like an awakening. Like if I take people out in the woods, and most people will never have like had the experience of like flipping a log over, finding an ant colony, and kind of gently picking through it and being like, look, here's where they're keeping the pupae, here's where they're keeping the larvae, and then you find that chamber. Usually there's like a chamber in that colony with
Hmm.
the queen, and she's surrounded by all the eggs, and there's often these small workers that are tending to her, and you can see this whole kind of society living in there. And in particular, this idea of like a queen and the workers, like um, I just remember being like 17 or 18, and I guess you like vaguely know about this your whole life, but like you read about it in books or something, and you start to really understand what it is, and it just totally captured my imagination. And so when I was teaching entomology that summer, I really didn't know anything about insects. I had never been taught about them before. I had some like teaching materials I was working off of and books I would look at from time to time, but I really had no idea what was going on. But I wanted to find a queen ant, and
Hmm.
I probably saw hundreds of them, but I didn't really know what I was looking at. So I never really was sure if I ever found a queen ant or not. But I remember it being like that idea of like the worker and the queen being like this very fundamental thing for me.
Yeah.
And of course the idea that it's like a superorganism where like the worker is like the skin cell or the somatic tissue, and the queen is like the germ line or the reproductive tissue.
Yeah, yeah, and you often like the canonical story that we're told as we grow up is like the queen's in control, and Napoleon's one of the symbols of his empire was a bee colony, which is also Hymenoptera like ants. But
Yeah,
that's you have the benevolent leader and then all the workers beneath it. But as you alluded to just now, like the deeper, more confounding take is that the queen's not in charge, the workers aren't in charge, they're rather orchestrated pieces of the same something. And that's
yeah,
really profound and heavy. And you wonder, oh, is the queen a slave to her workers? Are the workers a slave to their queens? There's all these interesting questions that bubble up all the way to the societal, socio-political level that we can actually get
for
from
sure.
just considering these fascinating little creatures.
Yeah, and
Please.
I don't know, because I was also reading E.O. Wilson at that time. We're talking about like 20 years ago, right? Like,
Mhm.
and I don't remember where I learned it, but definitely the the thing that sparked my imagination was the idea that the colony is acting as an animal. It's more like a collection of cells, and it's like an emergent property, and that
Yeah.
the queen is not controlling the workers or vice versa, but it's like a society. It's like an interacting unit, right? And I definitely that idea was definitely one of the things that made me become so fascinated with ants. It was never this other kind of thing of that there's like you think there's like telepathy or the queen's controlling that part. It's not really interesting. It's like science fiction. The real
Yeah.
thing is what's so amazing, right?
Yeah. The
Um,
orchestration. Yeah, well, in your master's and PhD program, were there any particular experiments that really set you on your course of further research or important discoveries that you made or that you're proud of?
well, so yeah, so I would say like just if we're sort of talking about how my career got going, I definitely had this like nascent interest in evolution and then kind of insects, and then kind of ants in particular among insects. And it was kind of in that order, you know. And uh, I sort of just had this lucky break of working in a research lab as an undergraduate. So I actually did my undergraduate at the University of Georgia, and I'm now going back there 15 years later to be a faculty member, which is pretty cool. And I had this lucky break of working in this research lab of a man named Ken Ross. And you know, I had never been exposed to like world class like scientific research before, right? I mean, you read about stuff and things, but like I'd never like met like a real scientist or anything, you know. And um, Ken is an incredible scientist who had made throughout his career a series of very, very consequential discoveries about, particularly about ant genetics and how genetic systems can sort of drive and the understanding of genetic systems can explain how they've evolved, you know. Um, and so it was really working with him that took that kind of nascent little interest and turned it into this like total passion, you know. Um, and his big discovery, he made a number of major discoveries, but one of the sort of central ones was that in Georgia you have these fire ants, and you'll have if you look closely, if you're paying attention, you'll see that there's like two types of fire ant colonies, some with a single queen in it and some with a few hundred queens in it. um, and there was some kind of early evidence that existed when he started working on this, but he fleshed it out a lot more, that it's really like two forms. It's not like a continuous spectrum of how many queens you have. It's like two types, like the one queen type and the few hundred queen type. And these two forms are extremely different in their biology. Like the one queen type are more like normal ants, where the queen is the mother of all the workers, and they're territorial. They form these mounds where all the ants in the mound are, or the pile as they would call it in Georgia, are the daughter of that queen. And they'll have a territory, so they'll kind of patrol a region that's like the region that they control. And if other ants of the same species come in, they'll have like fights over it and stuff, right? Um, Very typical ant behavior. But then there's this multiple queen form where there's hundreds of queens in the colony, and now you have basically no boundaries between colonies at all. So the workers are not necessarily the daughters of the queens, because there's tons of queens in there. I mean, all workers are daughters of a queen, but not necessarily the queen she's interacting with, because new queens can enter the colony and become reproductives. And the colonies don't really have boundaries. You'll have many different nests all right next to each other. And the workers can kind of freely exchange between them, and they won't fight. Um, and so uh, this it's not just the number of queens in the colony, but it's like this profound biological distinction between these two ways of living, like these two very different types of social organization. Um, and Ken's major discovery, to everyone's surprise, is that these two types are almost analogous to like sexes. It's almost like a male and a female in a sense, where um, there's a single chromosome that if you inherit this chromosome, it confers the multiple queen type. And if you lack this chromosome, it confers the single queen type. And that one genetic element, this complex chromosome, inheritance or not of it, is actually the sole determinant of whether they organize in this one way or the other, you know?
Yeah, yeah.
Um,
Do you mind if I ask a question about that? Um,
mhm.
are these, post discovery, are these considered as two separate lineages? Can they and do they intermix at all, or is
No,
it
they do, and that's what
some
makes them
okay.
like sexes, right? Is that they're
Mm.
members of the same species. There's total gene flow across the whole genome. There's just this perfect determination of whether you inherit uh, either you lack this novel chromosome or you inherit it, and that
Is there a
will.
dominance, like in sexes, if you have, the double X or XY, it's going to be totally determinant. When that
Yeah.
recombination happens, is there a Mendelian effect where if you have a little bit of the polygynous or multiple queen chromosome, you're going to be a multi-queen, or how does that
Mhm.
work?
Yeah. So uh, in this case, what we have is like this chromosome that we call social B. B doesn't mean anything. It
Yeah,
just
the
comes from
FB, right?
the history of how it was discovered. But um, so big B is going to form single queen colonies, and then big B little b is going to form multiple queen colonies. So the little
Is
b
that in
allele.
the grammar of Mendelian box genetics, the BB, B little b? Okay, that makes
Exactly.
sense.
Yeah, and so that little b allele is dominant in conferring the multiple queen form. You only need one copy of it to to have that multiple queen form, and it's kind of like a mutant form. So the big B is more like an X chromosome, whereas the little b is more like a Y chromosome. It's like the
Interesting.
weird one that induces the weird thing, just the same way as like in humans, developing female is kind of like a default state of growth, and then
Yeah,
developing male is something that's induced by this like weird broken Y chromosome that induces it.
yeah.
It's a lot like that actually.
So of course, we might be kind of jumping ahead into your research, and maybe you'll feel inspired to bring up certain graphics to explain some of these ideas. But one of the questions I have is, okay, you identify that the chromosome induces this large-scale, observable phenotypic change between polygon, multiple queens, and single queens, and then you ask, okay, what are those changes, and why does it cascade into these very observable effects? And
Yeah,
if I remember from your research correctly, one of the things that happens when you have this social gene that induces multiple queen behavior, it has to do with olfactory recognition.
that's
And
right. Yeah,
yeah, and you talked about how in the big queen colonies or many queen colonies, there's a lot of intermixing. Foreign queens can come in and establish themselves within the colony without being attacked, as opposed to single
yeah.
queens, which are very territorial. So maybe you could talk about the role of nestmate recognition, what we know, what was known in the past, what was overturned by this research.
For sure. Um, so this discovery of this genetic regulation of single versus multiple queen colonies, um, it turned out to be like this magic well of further discoveries, right? Once this genetic element was identified, further study of this thing led to all kinds of major and unanticipated findings, right? And um, the key observation at the beginning was that um, in these multiple queen colonies, all of the queens will carry this big B, little b genotype. So they'll never have the big B genotype, but they'll also never have the little b genotype. Um, and this is because the little b genotype is actually lethal. So it's a recessive lethal gene. So it dominantly confers this multiple queen behavior, but it recessively confers death. Um, but then the absence of the big B queens was not obvious why that would be the case, because if this colony is accepting new queens, you would think it would accept queens of both genotypes. Why would it only accept queens of this one genotype, right? Um, and so it turned out that this is because of a really interesting behavioral effect, where the queen, if she has that big B, little b genotype, she has a different pheromone profile. There are hydrocarbon pheromones on the surface of her body that are detectable to the other ants in the colony that are present in this big B, little b queen, but absent from the big B queen. So that little b chromosome is inducing a change to the pheromone profile of the queen.
Yeah. Can I ask a
Right.
question about that? Because I follow like Suzi Lab at UC Berkeley, and they do a lot of work on hydrocarbon profiles of ants, like the Argentinian ant. And
Mm-hmm.
one of the things that they discovered or show is that the variation between individuals in a colony is greater than the variation of hydrocarbon profiles between colonies. So, but
Sure. Yeah.
despite that, the colonies are still able to recognize themselves. I know there's some
Mm-hmm.
controversy on the methodology of like how do you actually measure lab mate recognition in lab. Um,
That's right.
but yeah, putting that aside, my question for you is going to be: have you guys, or are you familiar with research that quantifies these hydrocarbon changes? And is there actually an introduction via this specific chromosome of a specific hydrocarbon that is totally absent from another? Or is it another instance of this sort of murky hydrocarbon differences that are kind of different but also peak and are in the same areas, but yet somehow these ants are able to sniff out the recognition?
Yeah, that's the right question to ask. And the answer is that yes, there are hydrocarbons that are specific to the big B, little b queens. Um, and there is experimental evidence showing that um, by adding or removing those hydrocarbons from a queen and leaving the rest unchanged, you can change the behavior of workers toward the queen. You can change how they're perceived. Um, but it may not be the simple story that we would like, where there's simply a molecule present when you have the little B chromosome that's absent when you lack it, and that one molecule is necessary and sufficient for the whole behavior. There's probably um, a lot of complexity going on, and um, it's a whole other wormhole. But uh, yeah, Ken called this the bugaboo of chemical ecology. The fact that a thing that we call a pheromone is not often a single molecule, but it's more like a bouquet of different things um, that are perceived in a kind of binary way. But what's being perceived is not binary at all, even though the behavioral response to it might be. And so the queen pheromone profile, I mean, that sort of gets to the point that the queen pheromone profile is only half the story, because in order to have these different numbers or these different genotypes of queens in a colony, you need to have the worker ants who are actually doing most of the work in the colony behave differently toward queens of different types, right? You have the pheromone, and then you have the individual who perceives and responds to the pheromone, right? Which in this case would be the worker.
And all
And
this information comes from this chromosomal difference, the queen's expression and also the worker's reception and behavior change. It's
for
fascinating.
sure. And so the really amazing discovery was that um, because these queens, their genotype is big B, little b, um, if they mate to a male who has the big B chromosome, they can have daughters who have the big B genotype. So some of their daughters, the workers in the colony, will not carry this mutant chromosome. All things being equal, maybe you'd say half of them will and half of them won't. Um, and what we found is that they do something where when you introduce a queen of the wrong genotype, so a multiple queen colony, all of whose queens are big B, little b, you give workers from one of those colonies a big B queen. And what Ken found is that the workers in the colony will attack it and kill it. But if you look at the workers that are attacking it and killing it, it's only the ones that have the big B, little b genotype inside the colony. So the workers are a mixture of both genotypes, but it's specifically the ones that carry the little b allele that are the ones that are attacking the queens
Yeah.
that lack the little b allele.
Yeah, and if a core mechanism in this behavior and recognition behavior is the olfaction, the fact that there seems to be some sort of innateness based on the chromosome you have to the behavior that you'll express, and it's because there is the theory that a lot of this nest egg recognition is like what you're born into, so to speak. If you're an ant that's born into a colony and you just your queen smells like this will be the phenotype, or excuse me, the hydrocarbon profile you recognize. But here you have this interesting instance where they introduce this queen. Presumably, it's got the smell of the colony on it, but there's something in that hydrocarbon profile that they're programmed to react to. Really, really
That's
fascinating. Right. A whole wormhole or rabbit hole, termite hole to go down. Yeah. But I know
and so I'll just say that this really, and there's many more discoveries that have come out of this thing, but you asked about my work, and that was one of the things I contributed to, was this pheromone side of it. But you kind of don't want to miss the forest for the trees, because the big picture is just that this was the first time that we had evidence of like a genetic variation that's conferring a large scale difference in the way that a colony is organizing, the way that ants are behaving. And that the way in which that genetic difference induces this change might happen in part by the small scale, pheromone production and perception and stuff. But the big picture of it is that you have like the first time where you have like real evidence of basically how ants evolve, like how you go from one type of ant to another type of ant within a species. How do you make that jump, right? Um, and that kind of discovery, the fact that we can actually see evolution in this way, really kind of set the basis for the rest of my career, as you alluded to earlier, of like looking for um, these kind of large scale genetic effects, like genetic elements that can really induce major switches between like type A and type B. We sometimes call this Mendelian genetics, um, as opposed to what we call quantitative genetics. So basically, quantitative genetics being like something like height, where how tall you are is partially determined by your environment, but also partially determined by the sum of many, many genetic elements all throughout your genome. So it's sort of a totally normally distributed, continuous trait. Whereas something like eye color, we consider a Mendelian trait where maybe other features of your eye are continuous, but just the color of your eye has a simple Mendelian inheritance of
Yeah.
genotype that determines it.
Yeah.
And so when you
Punctuated
see this,
equilibrium.
right, and the amazing thing is that what people would say is that something like eye color is like, it's cool that it's Mendelian, but it's like maybe not really that relevant, right? Like it's not like it's helping you survive or it's really associated with your fitness in a major way. Um, and there's kind of this general, there has been historically this general belief in evolution that while these Mendelian traits, this kind of particulate inheritance, is at the bottom of everything, and it's good to know that that's how it works. It's not really the right way to think about evolution, because it's going to be the sum of many of those small things that's actually going to lead to changes in traits and actually cause animals to be sort of meaningfully distinct from each other, right?
Yeah.
But that's one view of evolution. But Ken's example is showing a very different view, that no, you can have one chromosome that really does induce a large scale shift in phenotype from one form to another, right? And that is like it's a much more kind of jerky, like large step way of thinking about evolution than was historical. And partially because of this historical belief, Ken's findings, a lot of them were not, people recognized that it was real and they believed that it was true, but they really heavily dismissed its generality for decades, until we have so many more examples of similar types of things happening, where now it's pretty much undeniable that this is a real force in evolution.
Yeah. And
Does that make
this
sense? Kind of lay
yes.
out?
And yeah, totally. And I'll try to bring it home for the listeners, if there are any, and to bring it into the relevance of your future research. But this family of effects that was begun with Ken Ross's research and then extended out, generalized out, is that what we would call supergenes? This idea that there's large scale transfer of associated genes that are tied together, like on a chromosome, and that these can be inherited outright as a group, and then those induce observable phenotypic and genotypic effects. Was that what Ken Ross's core discovery was? The supergene? Is that where the idea of supergene came from? Actually, ants. I think we might have lost our guy. Buck's back. Dan, you have a little hand up. Does that mean you want to speak?
When the time is right. Okay, you're back though. Continue.
Okay, Buck. Sorry, you cut out. I don't know if you heard. You're back. You're gone. Little B, Big B. It's killing me, dude. Oh, it's called Little B. It has nothing to do with bugs.
Yeah, everyone knows about Little B. Not many people talk about Big B though.
Amazing.
Yeah.
All right, Buck. You're back for real this time.
I'm back.
Big
Yeah.
B. Big B's back.
Yeah, Big B's back. Okay. Well, I was saying. Here's my question. Ken Ross, his work on these large group of interconnected genes that are inherited, like on this chromosome, that induce these large effects. Were these supergenes discovered via Ken Ross's work, and then generalized out to other ant species, and maybe you can
Mhm.
shed light on this too, to other insects, maybe humans as well. Is that
Yeah.
the legacy of his work, the supergene?
Yeah, I mean, I alluded, yeah, that this discovery came with a tale of many other discoveries afterward, right? Um, so, um, right. So what we've been discussing is that there can be these two discrete phenotypes, and there can be like a discrete genetic basis that underlies them, not like a continuous genetic basis that underlies them, right?
Mm-hmm.
Um, and in principle, you might imagine that a single mutation might be able to induce that switch between two discrete phenotypes. Like maybe you break a pheromone receptor or you break a hydrocarbon synthesis enzyme or something, and that would be sufficient to give you this switch, right? Um, and when he first discovered this, probably for 10 or 15 years, that's more or less what they thought that they were looking at. They sort of didn't know what gene it was, but they figured that it should be some kind of like straightforward mutation. And this was part of the reason why a lot of people were skeptical of the finding, is because people said, these single and multiple queen colonies, it's a very sophisticated thing. They're different in a lot of ways. And I would think you would need to change a lot of genes to get that type of a difference, not just one, right? Um, and so it was, yeah, after a whole lot of work and technology moving forward and all kinds of serendipitous events, comes to the discovery that it's what you said earlier, what we call a super gene, which is to say that um, you have an interval of the chromosome that does not undergo the process of recombination when cells are being reproduced for reproduction. So when a mother produces eggs to make her daughters, normally you can recombine between the two copies of the chromosome that the mother has to make sort of new hybrid or chimeric chromosomes that the daughter will inherit. But these supergenes, they do not recombine across the interval of the supergene. And so the end result of this is that you get something that is very similar to a sex chromosome, where you have this large interval of a chromosome that's non-recombining, and because it's non-recombining, you don't need to just have one mutation that distinguishes that single queen versus multiple queen form, but this non-recombining chromosome could actually contain thousands or tens of thousands of mutations all along its length, and then that could change all manner of proteins in many complicated ways. And so that could explain why a single genetic element is capable of inducing such a large scale
Yeah,
shift.
I don't, Dan, I want to give you a chance to speak here. I do have a question about the mechanism of this recombination, but Dan's had his hand
Mm-hmm.
raised.
Okay, some short general comments, and then I want to take us back to Denver, 2017.
Let's do it.
Well, I have a lot of resonance with what you shared about your interests, like how do we study the evolutionary ecological patterns across systems? How do different generalizations and abstractions help us find similarities and differences amongst all of these natural history encounters? And that's kind of like the Kantian idea that there will never be Newton for a blade of grass. Like, biology is just a history. It's not even a science. It's just about the field work. So we can kind of talk about that natural history angle. And then there's these repeated origination events with some similarities and differences. Like, there's the dependent lineage system in the Pogonomyrmex that I studied in graduate school, which have just within the Pogonomyrmex multiple separate originations of these dependent lineage systems, and in other harvester ants like Cataglyphus. And then there's all these silver ants. So all these kind of cool similarities and differences, and then understanding how does that make small continuous incremental changes and these like big large scale updates.
Yeah.
And that's even happening within the like eusocial. Well, there's termites, and so it's like all these different like fun levels and nested stories that have similarities and differences. What's the role of mechanism in all of that? How does entomology integrate molecular biology, which is really just beginning? So now let us go to Denver in 2017 at the entomology meeting. So what about the work with the Cronauer lab? How did you get into the Harp Ignatius game, and then where did that kind of vector you out to?
Yeah. So yeah, and I'll just say that your point about the sort of generality of these things, I think it's this really critical piece, which is that um, yeah, you study an animal like these fire ants, and you work out, oh, that they have this two types of chromosomes and stuff, and that's just like it's like natural history. It's like just a thing that happened. It's something that these ants are doing. um, but with genetics, it can often be something that no one knew was even possible. Something that you show that they're doing that was considered to be not possible, not how evolution works, not how genetic variation works. And then once you know that it's possible and you see one instance of it, people can go check and they can see if it's happening elsewhere, right? And so what we've come to find is that Kent's discoveries are extraordinarily general, and that for reasons that remain unclear even to this day, whenever you see phenotypic variation in an ant species, whenever you see like a large scale difference between two types, everything that's been published so far has said that that's a supergene every time. So it's a major, major driver of ant evolution. And in fact, even Ken's supergene itself, that exact same supergene evolved independently, at least once in the silver ants, the desert cataglyphus ants. The same exact stretch of the chromosome induces the same exact phenotype, but the common ancestor did not use this chromosome to induce this phenotype. It evolved independently on this same chromosome in two different lineages, right? Um, and so that's the beauty of this type of study of evolution in natural history, is that in one sense it might not really be experimental, it might be very descriptive, but when what you're describing is something totally unprecedented, sometimes it can also turn out to be general and really predictive of many additional systems as well. um, but yeah, so I worked on this with Ken, and basically the issue was this lack of experimental thing, which was that we were studying these pheromones. We could describe these differences between queens. We knew that this chromosome was somehow responsible, but um, the big bee versus little bee queens, they differed in like 600 genes that were found on this chromosome, and most of those genes have at least one, if not multiple, mutations that maybe don't do anything, but could potentially be contributing to this difference in phenotype, this difference in pheromone profile. So figuring out the causality of how this chromosome changes the pheromone profile seemed to be basically impossible, right? Um, and so it was for this reason that I got really excited about the idea of doing genetic experiments, like taking animals into the lab and actually manipulating the function or the expression of genes in order to show experimentally that a particular protein or a particular mutation is actually causing a particular phenotypic difference, right? And how it does so. And so I decided to work with Daniel Cronauer in my PhD. He studies this organism called the clonal raider ant, Ooserea baroi. And at the time, he was in this very early stages of wanting to develop this as like a new model organism for ants, and in particular a genetic model organism, a species that you can keep in the lab and you can rear for long periods of time under controlled conditions, and you can do controlled genetic perturbations. And so my contribution to this was 4 years of backbreaking work to make the first CRISPR protocol in ants. So we uh, developed the methods to collect eggs and inject them, do sequencing to screen for injections, rear up, and basically generate mutant lines of ants where you've used CRISPR to introduce a stable modification to their genome. Um, and because of my background studying pheromones, there was a very obvious choice of which gene to go after, um, which was a gene that is required for the function of the odorant receptors. There are these receptors that are involved in the sense of smell, and in ants, there's a very large expansion in the number of odorant receptors that ants have relative to other types of insects. And so there was a hypothesis that these receptors are probably what ants are using to perceive pheromones. And so what's really cool about that is that this receptor family has a single gene called the co-receptor, and this one co-receptor protein is required for any of these receptor proteins to function. So if you knock out that co-receptor, you also knock out all of the receptors at the same time, and you can do a really clean test of when an ant doesn't have odorant receptor function, is it still able to perceive pheromones? What happens to its behavior, and so on.
Yeah, so presumably you knocked out. Oh, I actually read your research last night. So you used CRISPR to make these changes at the egg level, which is really cool. Knocked out these ORNs, these odorant receptor neurons, and then what'd you find? I don't want to spoil.
Yeah, so this was basically the first CRISPR ant, and it was kind of a proof of concept to show that we can do CRISPR in an ant, right? And we were hoping that some kind of interesting biology would come out of it, but in general, the hypothesis that odorant receptors are used to perceive pheromones felt all but certain, right? And so we expected that the ants would not be able to perceive pheromones, but maybe that wouldn't teach us a ton about biology, but at least it would be like a nice confirmation of this, right? And so that indeed happened. All the different pheromones we tested, these mutants were not able to perceive. In terms of the pheromones, it's a little bit of an ancillary point, but one really kind of cute and interesting point was that the mutant ants that lack the function of the odorant receptors, the most striking phenotype that they have is actually that they spend almost all their time wandering around outside the colony. And it led us to realize that there seems to be a very strong pheromone that's controlling whether an ant believes that she's inside the nest or whether she believes that she's outside the nest. And inside versus outside, their behavior is totally different. You know, it's like don't use your indoor voice when you're inside. Like when you're in the nest, you act one way, and when you're out of the nest, you act another way. And it looks like these ants, because they were not able to perceive this signal that tells them that they're in the nest, they acted as if they were always out of the nest. And that's pretty cool because this inside nest, outside nest pheromone had never been proposed to exist. I mean, lots of different pheromones were described from ants, but that there would be a pheromone for this particular thing had never been proposed before. It actually still remains not discovered, but very clearly from the evidence that such a thing must exist.
Yeah. One question on the mechanism. For when the ORN, you said that co-receptor gene was knocked out. What is the effect on the neuron structure itself? Does it just not get produced outright, or is there some identifiable catastrophic mutation?
Well, right, and so that's the next point I was going to get to, which is to say that with the exception of this nest thing, which is kind of cute and cool and worth looking into, for the most part, these pheromone deficiencies were what we expected to see, and maybe don't teach us a huge amount about like how pheromones work and things like this. But what came as a complete surprise is that when we opened up the brains of these mutant ants, there was like an 80% reduction in the size of the olfactory lobe. You know, the brain region that's responsible for the sense of smell was totally shrunken and underformed in these mutants relative to typical wild type ants. And it turned out that in addition to the brain region not forming, also in their antenna, in the kind of like the equivalent of their nose, the neurons that are involved in the sense of smell that would express these odorant receptors were all dead, all absent.
Were they dead or absent? They just weren't there, or were they atrophied?
No, they were. I mean, they had died earlier and then gotten cleared out, so they were both dead and absent.
So just like a hollow, for lack of a better term.
Yeah, it actually was like a hollow space in there, really bizarre. And what's amazing about that is that these experiments that knock out the function of the odorant receptors have been done in a number of other insect species, and this effect has never been observed before.
Yeah.
Um,
What does the knockout do? Does it perforate the cell wall or something? What's the actual catastrophic failure induced in the neuron? Is it
um,
microtubule based?
we don't know. I mean, we don't have a specific description of it, but um, the thinking is that the primary job of that neuron is to receive these olfactory signals and then have an action potential, like to fire and send
Mm.
information into the brain. And um, it has been described previously that when you knock out the odorant receptors, those neurons stop firing, right? Because the thing that's making them fire is the odorant receptor. Um, and so what we think is that um, these neurons and their associated brain region really depend on that firing to function, that they use the firing to wire where they go in the brain. And if they're not firing and they don't wire to the right place in the brain, they're basically going to do programmed cell death. They're going to do apoptosis because they're not their function cannot be put together,
If you don't
right?
use it, you lose it.
But that level of neuroplasticity, or you could call it receptor dependent survival of the neuron, receptor dependent development of the brain. Um, these are things that are typically associated with mammals and
Mm.
more complex organisms with larger brains. In fact, it's well known that things like this happen in mice and humans and dogs. But in insects, this type of activity dependent development, kind of plasticity, uh, had not been observed previously. And it was believed that an insect brain does not do things this way. An insect brain is instead, quote unquote, hardwired, which is kind of a bullshit term. It doesn't really mean anything, but it sort of alludes to the idea that it's like more stereotyped and less flexible, right?
Yeah, fascinating.
But here we're seeing that an ant brain is actually acting more like a mammalian brain in this
Or a
regard.
mammalian brain is acting a lot like an ant brain.
Yeah, fair enough.
Yeah, that's fair.
They
Okay.
might have been doing it first. Yeah.
Yeah. So, just in general terms, where do you see this CRISPR technology being applied to in ants and insects more generally now that you've done this work? And maybe just give us a little anecdote about why you described it as backbreaking. Why was it so hard to get ants on the CRISPR wagon?
Well, it was hard and it remains hard. I mean, genetics has always been a numbers game. The reason why fruit flies are such a good model system is because you can grow up a very, very large number of them. And um, ants are hard to rear. They form these large, complex colonies, and getting access to that high level of reproduction and viable embryos that you can manipulate, the amount that we can rear up in ants is orders of magnitude less than you can rear up in flies and requires significantly more labor to do
Yeah,
so,
because you were
right?
actually injecting CRISPR changes via needle into these tiny little eggs.
Yeah, and so it's just about rearing them up and fine tuning the protocols, and figuring out things like how to do the injection in the gentlest way possible so that the egg will survive, and all kinds of stuff like that. And those problems are slightly better now, but it will always be harder and slower to do this type of work in ants than other organisms. In terms of the future potential, uh, there was kind of a bifurcation. One thing that you can do is you can take the fact that you can do CRISPR and do these injections, and then you can build on that to do more complex types of genetic modifications. So I was a co-author on a paper last year where um, they introduced instead of knocking out a gene, um, they used the promoter of the gene that we knocked out, the odorant receptor co-receptor, and they used a transposon to introduce a new gene into the genome that's expressed in the same cells that express the odorant receptor co-receptor. And the new gene that they introduce is a genetically encoded calcium sensor. So it's a fluorescent protein that in the presence of calcium will become more fluorescent, and in the absence of calcium is less fluorescent. And if you introduce this into a neuron, those are the genes that are expressing the odorant receptor co-receptor. If you introduce it into a neuron, then the fluorescence of the cell will be an indicator of how active the cell is, because when it fires action potentials, calcium will come into the cell. And so you can use this technology to look at these odorant receptor neurons and actually watch, while an ant is alive and you present her with a pheromone, what kind of pattern of activity scatters across that region of the brain. So
Yeah,
there are
fascinating.
kind of further engineering applications of CRISPR and other things. But I played some role in that work, but mostly after we did this proof of concept, I actually made a turn, and the way I kind of felt about it was like, now that we have this in our back pocket, we know that we can do this. Um, and so what we really need is to have a better understanding of the biology to find genes that we can study that are going to teach us a tremendous amount about the ants. Because this odorant receptor co-receptor gene was kind of a magic thing where we had a very clear hypothesis, a really good experiment to do, but after that, it wasn't obvious. Like, we don't really know enough about ant genetics to know, like, well, what other genes will we want to knock out, and what other things will we want to test? You know,
Yeah. Dan,
you
you
have a
want to say
question?
something?
Yeah.
Yes. We are early in the transformation from entomology being stamp collecting on through including systems biology. And Tucker, please mute.
Oh, sorry.
And so, always learning from the fruit fly and other insect research lines, kind of piggybacking, lagging, but then leapfrogging, tandem running, if you will. There's a lot of amazing opportunities. Like I just saw an open source software package called Flybody that was released by DeepMind and Genelia Farms for fruit fly biophysical modeling. They have a full connectome. Honeybee has a full connectome. Like, these kinds of things start to come together, combining with multi-omics, and we could be very quickly going towards a place where we have more like mechanistic, causal, multi-scale models from molecular and cell scale all up through organizational at the colony level, and then ecological, and it's kind of like all going to be fitting together in this big puzzle with Bayesian multi-scale modeling rather than just one-off experimentation.
Yeah, and I think that one of the things you said that was really important to me is that, like, um, our experiments with ant pheromones um, was based off the fact that we have a really good understanding of how the olfactory system works in the fruit fly, and that in this case, actually, that understanding of the olfactory system in the fruit fly came from prior studies in the mouse that were meant to be useful to human medicine and stuff, right? So there was a Nobel Prize, I think, in 1994 for the discovery of the olfactory receptors and the structure of the olfactory lobe, and the basic details of how the sense of smell works, right? And then, so then we were just building on sort of decades of knowledge on this type of stuff. And that's great when you can do it, but it does pose a little bit of a paradox when you want to use the ant to discover something new, but you can only be doing that by piggybacking on these major fields of research that are decades old, right? And so that's part of the reason why, after we did this CRISPR, I felt like, this is good. It's good to know that we have this, but now I need to go and do the really hard work of trying to develop new knowledge that's coming directly from the ants and that's not taking a hypothesis from these model organisms, right? And so, in order to do that, my view has always been that there's basically two things that make ants different from the proficient hunter uh, solitary wasps that they evolved from. And one of them is that they have these pheromone communication and this really complex communication system that allows them to coordinate in these large groups. But the other major difference, of course, is that they have these morphological castes, like a worker and a queen. And so I always felt like, castes and pheromones kind of together, it's like peanut butter and jelly. Like those are the two things that together are the magic sauce of what an ant is. And feeling like we had a really good understanding of how pheromones work, my impulse was immediately to be like, drop all of this and now let's try to do that same type of thing with casts and figure out how that works as well.
Could you
Yeah.
share some pictures and tales from your recent fieldwork visit? Like, what was the context here, and then what was it like, or what was funny and interesting?
From the fieldwork I just did, or
Yeah.
so yeah, so I guess I'll just say that. So basically, I've been studying cast development and evolution for the last maybe 6 or 7 years, pretty much full time. And I'm really trying to dig into that and find these basic molecular mechanisms to get to a level of understanding similar to what we have with pheromones. And to do this, there's lots of different ways that you can do it, but um, I'm taking a similar approach to the one that I learned from Ken and that I've used in the past, which is to heavily rely on natural genetic variation. So a lot of my research, it relies on looking at the literature, looking at natural history reports and descriptions of species, and looking for places where you have polymorphism, like two very different types of morphology or two very different types of behavior. And then we can do some basic kind of high school statistics to guess if that polymorphism seems likely to have this simple genetic basis. And usually it does look like that. And if that is the case, we want to go out into the field and collect them in order to do sequencing and try to map the genetic element that induces the phenotypic difference. So in Vietnam, I was just basically pursuing this same workflow with a number of different species. I was in Vietnam for 6 weeks, which is pretty cool. Um, and I looked at multiple different projects. One of the most spectacular of these is this ant species called Acanthomyrmex. Do you know Acanthomyrmex, Daniel? Man, this thing
Yeah.
is
If you
nuts.
have photos, please share.
I'm trying to see. So I might just pull up um, a paper. Let's see.
Yeah, take your time. It's worth it.
Briefly,
Yeah, let's check this out.
just for a footnote for future times, Tucker came out several times to me to help with my field work in Arizona, New Mexico.
Yeah.
So that was sort of some of our early Genesis fun moments, but like kind of weaving
6
from
week
this, the molecular work, the computational work, the field work, the inner work. Really awesome how in this short time you're just weaving these pieces together. So
For sure. So let's see. I got to get you a better photo. Um, that's perfect. Okay. So here's just a photo from. Oh, it's actually a. Jesus, it's a YouTube video. Here's just a photo from Google to get us started, and then I have a paper I'll show you.
Yeah, this is
we want to have unified databases. Just do generative AI.
Wow.
So
Beauties.
So
the brain bug.
that's the worker, that's the soldier, and then that's the queen. So it has this wild level of polymorphism between the three, where basically the soldier, the way I put it is that like 2/3 of the mass is just going to the head by itself.
For the muscles, I presume it's jaw muscle, right?
That would be the thinking. Yeah. So let me actually see if I can do something. Um, my
Yeah, never
my
seen an ant like that.
So if I put on my desk view camera, okay, good. So that's my phone. Um, Let me try something here. So
Picture in picture in picture.
I have a whiteboard table in my living room, like a real scientist. So
All
can you
right.
see? Yeah, okay.
Yeah, yeah.
So basically, okay, this is mirrored, but I don't need to know that. Let me just do dot, uh, dot. Okay, now I know where I need to be writing
Oh
between
goodness.
this and that. Okay. So basically what you have is in a typical ant you have a relationship. Oh, but in the well the does the plot backward for you? Or does it look normal?
It
Looks
looks
good.
like a
Looks
normally
good.
faced L right now.
Okay, it's good. So basically what you have is if we have body size here, then we basically if we have like a histogram, so if we have frequency here, then basically we have the worker, the soldier, and the queen. Does that make sense?
Yes.
So then we can put on this, this is body size. We can have a separate y-axis that's um, the head width, okay? And now this second y-axis is going to go like this. You see?
Yep. I
So
believe it.
basically, the queen is bigger than the soldier in body size, but her head is smaller than that of the soldier, right? And what we think is that there's a simple explanation for this, which is that um, you can have a pie chart of uh, the fraction of the body that goes to the head in the worker. Then you have a bigger pie, but a huge fraction of it goes to the head in the queen or in the soldier. And then in the queen, you have the biggest pie, but then you're back down to like a smaller fraction going to the head, like this, right?
Hmm.
And we think that the reason why the fraction of the head goes down in the queen is because there's another slice of the pie that's basically zero in the worker and the soldier, but that's very large in the queen, and that would be the wings and the ovaries.
Hmm.
So basically, because the queen is growing these large wings and ovaries, her head is reduced because it's taking energy that was free to go to the head in that big soldier. That makes sense?
Yeah.
So this is a really extraordinary species and an extreme example of path polymorphism on its own. But what's even crazier about this ant, I mean, kind of unbelievable, honestly, is that um, we also have in this species what I would call a worker-sized queen and a soldier-sized queen. And we also have something that's called an ergatoid queen. um,
Amazing.
so uh, this one species has four types of queens in one species. Um, and with that, I'll switch my camera back to my normal camera. Um, but so basically, you're looking at a species where you got this worker and this crazy soldier, and then this is a normal queen. But sometimes the colony will have a shrunken. They call them dwarf queens in the literature, and in my prior graph, I was saying size. What I really meant was like overall body mass, right? Where this queen is clearly more massive than this soldier. Even though her head is smaller, her overall mass is definitely bigger, right? But there are other queens you'll see where their mass is exactly the same as the soldier. So that's that soldier-sized queen. And then there's other queens you'll see where their mass is exactly the same size as the worker. And then there are these ergatoid queens where the mass is bigger than a soldier but smaller than a regular queen. So they're intermediate. And those ergatoid queens will have partially formed queen tissues, but not fully formed queen tissues.
What
Yeah.
what did you
And
do
if I
or
could please,
collect? Like, were you doing
So
behavioral assessment, morphological measurements, collecting samples for omics, or what?
so for this species, it's been reported in the literature that there are these 4 types of queens in one species. And an ant that has 4 types of queens, I mean, there's no other example of that. I mean, I would know, right? I can't think of any example. I mean, you only ever hear of species that have 2 types of queens, like bigger ones and littler ones. That exists. And even that's rare. Most species only have 1 type of queen, right? But sometimes they have 2. But I don't even know of an example that has 3, right? This has 4 types of queens, and they're all discretely different, all in the same species. And that's it. That's the extent of our knowledge, just that they exist. And there's a paper showing that the ergatoid queens really are queens, that they can reproduce and be the sole reproductive of the colony and so forth. Um, so the first thing to do is just to try to find colonies with all of these different queen types in them and use some simple kind of like paternity tests, like genetic fingerprint data, to see if all of these queens are reproducing, can reproduce, and if the workers are daughters of these mothers, right? An alternative possibility is that maybe these miniature queens, like the worker-sized queen or the soldier-sized queen, might be something like a parasite, where it's taking over the colony and it's not able to produce workers of its own, but instead it's just hijacking the workers of another queen and then using that workforce to make new queens that will go off and found new colonies.
yeah, fascinating. So
But
they
it
have
remains
multiple
unknown.
queens. They have many queens in one colony as well, and of all these different phenotypes.
No, so when you have the big queen, can you still see my screen?
Yeah.
When you've got the big queen, you're only going to have one. But if you have these dwarf queens, then you're going to have multiple of them.
Hmm.
And yeah, and here's the paper. I realized that it's not. Oh wait, this is wrong. Oh Jesus, yeah, this is actually something I wanted to discuss today. By the way, this is
Okay.
a hard pivot. But Daniel, I almost considered canceling because this is Trump's feud with Harvard. My grant was canceled last week. That they just unceremoniously canceled my grant, along with all federal grants to Harvard for absolutely no reason.
All federal grants, so just
Yeah,
no more money coming in the door.
yeah. This award was terminated. It has nothing to do with me. I didn't do anything. It's a grant that I won from the NIH, and they just said we're not giving any more funding to Harvard because just for pure political reasons. There's absolutely no reason for it at all, but it's thrown everything we're doing into complete chaos.
Wow.
You know, horrific, absolutely horrific. But so we will talk about that in more detail later on or another day. But here's the pictures of these wild things. So um, that's that typical winged queen there, and then this is that dwarf queen. So this one is the size of a soldier.
Mm-hmm.
Um, and then there's another dwarf queen, maybe in the next figure.
Great photos.
Yeah, there she is. That one's the size of the worker, right? But
Yeah.
this one, that's the full-sized queen. And you can see that the scale bar might change, so it's a little bit tricky.
Mm-hmm.
That's the full-size queen. That's the one the size of the soldier, and then that's the one the size of the worker.
Wow.
And then there's a fourth type, which is not reported in this paper, but it's uh, intermediate in size between the soldier and the regular queen, and it doesn't have wings, and it's what we call an ergatoid queen.
Wow.
Um,
Do you think
so
there's any super genetic connection here between these different phenotypic expressions?
Yeah, I would bet my life on it, dude.
Okay, okay,
Got it.
bet your grand on it.
Well, yeah, too late. Uh, yeah, I think that, I mean, there's lots of different ways it could play out, but it's just an extraordinary place to go looking for something because nothing is known about this, other than that it exists. And I found it. I found both types of dwarf queens when I was there collecting.
Wow.
I didn't find any of the ergatoid queens, but I think they occur in maybe Singapore or China based on stuff I've seen. And so, but it's all more to look into. But, a really powerful tool to try to understand how cast development works is to find these species with these wild polymorphisms in different types. And then, yeah, in all likelihood, there will be some type of genetic basis for these different types of queens. We can map that. We can look at what genes are being expressed, and we can really try to figure out what is this underlying system.
Fascinating.
Yep. So many amazing natural histories that can be used to trace back from phenotype and to give this sort of opposite direction from forwards and reverse genetics, and where else to work backwards from the most amazing, beautiful, multi-scale hardware, software, wetware, collective creatures that creating a toolkit, a pattern grammar, software, multi-omics, like to be able to understand the ant colony in context and have tissue specific gene expression all the way up on through like multi-agent simulation. Like that is the kind of technology that transfers across so many different species. And it just
Yeah.
makes me think about learning about ant behavior, where we're always thinking about amongst nestmates and about the hormone influences on behavior, but also the environments, like that kind of behavioral complexity, collective behavior situation, and thinking like, wait a minute, I mean, people are studying behavior in all species collectives, yet they don't call it collective behavior. And so like the ants field gives us these multi-agent views. So it is hard to convey that it is kind of like the uber tech of modeling complex adaptive biological systems. Which,
Well, and I'll give you an example real quick, which is to say that I just showed you that crazy species, right? Um, the reason why I'm interested in things like that is because of this. So here we have a picture of a mutant ant that I discovered. We call it a queen-like mutant. So this is our clonal radar ant species, the species in which I did the CRISPR. And basically, what you have here is a wild type, a regular one, and what you here have is this worker-sized queen. And on this next slide here, I'm just going to show, I don't need to go into any detail about it, but basically, um, in most species, very close relatives of this one, that queen is always bigger than the worker. But in this case, we have this mutant that arose where the queen and the worker are the same size, right? And I've had this mutant for like 6 years. I mapped the mutation. I found a super gene in a social chromosome that's almost certainly responsible for the trait. Um, and in that super gene, I found some enzymes that appear to be broken. The splicing is broken of these enzymes, and the function of the enzymes is unknown. But bioinformatically, they're predicted to be able to bind steroid hormones, including ecdysone, the most important steroid hormone of insects. And they're specific to the fluorescent hybridization shows that they're only expressed in the hormone synthesis gland of that hormone. Um, and we actually took these, I won't show you the data, but we took these enzymes out from the ant and put them into a fruit fly, and I can get a fruit fly to basically have morphological casts. So I
Wow.
can get
Good.
it where
Finally.
the big fly grows normally. There's no this is a magic thing. You overexpress this enzyme. There's no phenotype in a well-nourished fly, but you starve the fly and it has disproportionately small wings. So under the condition of starvation, the wing growth is reduced, which is exactly like what's happening in ants, because this enzyme that we found is specific to worker ants, and our mutant that resembles a queen appears to have this enzyme broken. It's
Yeah,
not able to express it.
and I think
Um,
so many of these mechanisms are going to play into core like hormonal neurotransmitter, like just
that's my point.
body fat and like the forager phenotype, which is like more
Yeah,
of
and
all
so
those kinds
my
of
point
things. Yeah,
is, so we went a little bit out of order here, right? But we talk about supergenes, and then um, here's an example of mapping a supergene, and it traces us back to this pathway, right? And ecdysone is the insect version of estrogen, right? So this is not some insect specific thing, and I think that the pathway that I'm hitting here is probably a pathway that couples organ growth with body size, which is a really fundamental thing in all animals, that your body size can be a little bit flexible during juvenile development, and you need to flexibly adjust the size of all your organs to match it. The mechanism that mediates this is completely unknown. We do not know what is that integrator, that global mechanism that couples organ growth and body size, even in humans or mice or fruit flies. But here we have an ant where the coupling of organ growth and body size is all fucked up. We map it back to the insect version of the ecdysone or the estrogen pathway, and then we put it in the fly and we mess with the organ growth body size coupling,
Wow.
which really suggests that we might be hitting this pathway that would be present even in flies, maybe present even in humans. But it's what pathway we're hitting that this does, we don't know. It's somewhere in this steroid hormone, estrogen related kind of area, but there's a lot of different things to work out.
Yeah. I
And I
mean,
just wanted to put that in at the end just to say that that acantha is like a crazy thing with these crazy little mutants and stuff. But then where that can take you, it could be to medicine, right? It could be to something that's actually relevant to humans, or just to a much broader understanding of biology. It's not just a trivial thing to work something like that out.
yeah. I want you social fruit flies by 2028. We just might
Well,
need
yeah, and
them.
it's just you mentioned, Daniel, that the idea of this engineering and learning from these and using them as forget the word you used, machine or something.
yeah. I mean, the nexus, to give just this narrow example between sex slash caste slash phenotype contextual hormone production and receiving, and the way that that tunes into the way that generalized hypertrophy and nutrient limitation relate to allometric growth of different tissues. I mean, like, that's the difference between a meal giving healing and supporting a healthy body composition, and like regeneration of organs and limbs and
Yeah,
flexibility with developmental differences. um it's like that's an incredible nexus,
yeah,
and you're so right, it spins off into our fundamental understanding of the evolution of systems and these directly applicable methods and candidate hypotheses, because it's the conserved mechanisms like the insulin signaling, these kinds of things that are
yeah,
very like durable over evolutionary time, that end up being important targets to pursue in health.
totally. Yeah, and one thing that's really exciting about this body of work that I'm alluding to is that our mutant, we found this enzyme. The mutant is developing as a queen, and they're not expressing the enzyme, whereas the wild type expressed the enzyme at a high level and it's developing as a worker, right? And then we put it into a fly, and under starvation conditions, we get that same thing, right? Um, and furthermore, the mutant has um, wacky levels of ecdysone. They're up at some points, but down at others. But it's totally dysregulated, the level of ecdysone it has, um, and there are all kinds of genes that are annotated as being involved in steroid hormone signaling that are differentially expressed between the mutant and the wild type. So the point is, and our key candidate enzyme is specifically expressed in the ecdysone synthesis gland, right? So the point is that there's a clear story here pointing straight at ecdysone, right, as causing this. Now the problem is that ecdysone has never been described to have this kind of effect, where it just affects the growth of the tissues, but without affecting other things in the body. Typically, if you had a higher level of ecdysone, that would be expected to lead to pupation, exit of the larval stage at an earlier time and a smaller body size, or vice versa if you had less of it. But our mutant is the exact same size and has the exact same developmental timing as the wild type. And in our fly perturbations, the same seems to be the case, right? So despite the fact that everything points to ecdysone, our phenotype doesn't match ecdysone. Furthermore, our mutants
Hmm.
have differences in ecdysone levels, but when I feed them the ecdysone, I can't recapitulate the phenotype just by feeding them the ecdysone alone, right?
Wow.
And finally, and this is kind of the nail in the coffin, is that even though the genes that are differentially expressed are overwhelmingly annotated as ecdysone associated genes, um, there's no signature whatsoever that the canonical receptor of ecdysone, the ecdysone receptor, is being differentially regulated between my wild type and my mutant. So all of the upstream targets of that canonical receptor are expressed in the exact same way in both genotypes. And there's also no enrichment of the binding site of that receptor among the differentially expressed genes. So basically, what this implies is that we're looking at something that looks like ecdysone in every way, but doesn't act at all like ecdysone in any of the ways you would expect, right?
Hmm.
And that means that it's very plausible that it's not ecdysone, it's actually maybe another steroid hormone. I mean, at the moment, there's only one steroid hormone that's known to be made from this tissue, which is this insect version of estrogen. But maybe there's an insect version of testosterone or some other steroid hormone that we just haven't discovered yet. Or alternatively, maybe it is the ecdysone we're hitting. Maybe ecdysone has two receptors. One of them mediates the canonical effects on growth and developmental timing and body size, but maybe it has another receptor with non-canonical effects on how big the tissues are and the relative size and coordination, right? Which I just brought that up, Daniel, because it's basically what you said a moment ago, right? About um, the possibility that this type of fundamental knowledge could mean the difference between giving a drug treatment that induces a cancer versus induces regeneration, or there's a major problem with menopause where you can give people progesterone and you can advance them through menopause and really alleviate a lot of negative symptoms because the period of menopause is way shortened by it. But it also elevates the cell division rate and leads to a small but meaningful increase in cancer risk, right? Um, but if you could give that progesterone while also giving an inhibitor of whatever receptor mediates that cell division, then this therapy would be viable in a way that it isn't currently,
Yeah.
right? And that gets at this idea of like two sides to the same hormone or multiple kind of ways that it can be signaling, right?
Once again, bugs
But
prove to
yeah,
be incredible teachers.
Buck, this has been very great to discuss. Just to respect the time and where are you headed? Where
Yeah.
do you want to see the path unfold for you and the colony?
So so basically this whole pitch I just gave you about ecdysone and the coordination of organ size and body size, Daniel, you and I talked about this right when I was on the beginning of this work, right? Which was like 8 years ago. And this has been my obsession for that whole time. And I've been really, really trying to advance our understanding of this. And we have a couple big papers coming out this year that I do think are going to mean pretty significant progress for this problem, right? And so I'm pretty happy with what we've accomplished. But it's also been really, really hard work. I mean, backbreaking work to do this molecular biology and stuff. And um, the rewards come really, really slow. And I got kind of burned out and discouraged. And so I'm taking a little bit of like a sabbatical year right now where I'm officially starting at Georgia in August. but I'm not actually starting on campus until March. And right now, I'm at Harvard, basically working on packing up my lab and stuff. But I already did this 6-week trip to Vietnam. I'm doing a trip to Finland just in a couple weeks, and then it'll be Japan, then Argentina, and then one more that it remains to be seen. But basically, all of these trips are to collect samples for sequencing, population genomics, stuff like that. But also to kind of rest and recover and get space, find the time to finish these old projects and get everything published, and to try to kind of get my balance back in this hellish scape that we're in, you know. And so that's basically what I'm doing right now, and something I felt like I needed to do for the last few years with all of how stressful and hard everything has been. But especially, I mean, I'd been planning on doing this trip for the last like 2 years and slowly putting the pieces together and stuff. But now it seems more important than ever, right? I mean, um, they just canceled my fucking grant for no reason. I mean, it had nothing to do with me. I didn't do anything. And
I could have
in a
stepped out there.
sense, I don't even really work for Harvard because the grant pays my whole salary and the salary of everybody in my lab. And I wouldn't still have a job at Harvard if I didn't have this grant, right? Um, and they just, because of some weird political argument, they just canceled it last week, right? And it just shows, I mean, my partner had a fellowship that was classified as a DEI fellowship that got canceled. Um, and you know, we're really under attack here, you know. And I think it's worth being very clear and honest about what's going on. I mean, this is fascism. This is like when you have the color revolution and you drag all the academics out into the town square and assassinate them, you know. um, and it's not to that level yet, but it has rumblings of that. And it just makes me even more glad that I'm taking this time to kind of get my balance back and get my strength back, because I think we're really going to have to fight for what we believe in, and to keep going, because there's really strong forces that are trying to stop us from doing science and stop people from actually caring about what evidence says and what the truth is. And, you know, if they succeed, people will die. We won't get medicines that we could have had. We won't get agricultural treatments that we could have had. We'll have horrible economic policies that lead to unnecessary poverty and death, you know. So it's not an acceptable thing to let that happen.
Yeah, well, hopefully your grant situation will be more amenable and down south, and you can continue this awesome work. And the collection trip around the world sounds like a fantasy. It sounds amazing. Please keep us updated, and maybe we'll have you
Yeah,
back on for episode 100.
for sure.
Buck, you shared truly so many important pieces. I really appreciate that. Again, we could weave it back together. I resonate with what you've said, and I wish you the best, and we at Talktoptera support you fully.
Cool. Thanks, y'all. It was great talking to you. I really appreciate
Nice
it.
talking to you. We'll reconnect soon.
Thank
Bye.
you. Peace.
All right. Peace.